Okay. Well, my name is Carla. I'm on the Mersorcium board. That's why here's our logo. Um, I'm gonna present the last paper, a paper that has been recently published, called Unmasking the Physiology of Mercury detoxifying bacteria from polluted sediments. So I'm gonna do a very small introduction, um, because everybody basically knows all the basics needed for this talk. So this is the Mercury cycle. I'm not gonna explain the whole thing, just remark that we're gonna be talking about sediments, specifically demethylation and reduction of methyl mercury and inorganic mercury in sediments. These reactions can be biotically mediated, and this is done through the MER operon. So a quick overview of the MER operon for those of you who don't usually work with demethylation. So the operon has two regulators. One is MER-R, which basically starts the transcription, and one is MER-D that stops it. Then we have a bunch of different transporters that maybe or maybe not, they are present on the operon. Some of the transporters, most of the times MER-C, MER-T, or MER-P, can even be present without the enzymes. And then we have the two enzymes that are organomercurial liase, which is responsible for the demethylation of mercury, coded in MER-B. And we have the mercuric reductase, which is responsible for the reduction of mercury. And is coded on the gene MER-A. So the principal objective of this work was to find the best candidate for bioremediation and also the best conditions for mercury bioremediation in polluted sediments. So in order to do that, we went to 3 sites, 3 areas, and collected fresh sediments. We selected 2 areas in Spain, one in Basque Country and one in Catalonia, in Barcelona, and one location in Sweden. So we selected these 3 areas and collected different samples in each area. They have different concentrations of mercury. So the Bay of Biscay Bay, we collected 8 sites that went from 0.5 ppms to almost 7 ppms. In Barcelona, and this is, the Biscay Bay is contaminated due to a chloralkali plant. In the Barcelona site, we selected 2 places with different concentrations of mercury, up to almost 2 ppm, and this is mostly just an urban area. And then we went to one place in Sweden. I'm dyslexic, so I'm not even gonna try to pronounce this word, but you can all give it a try if you want to. And this has a very, very high concentration of mercury, almost 20 ppm. So from all of these fresh sediments, we used different mediums and different methods of isolation to get the MERCLAB culture collection with more than 2,000 isolates and more than 500 consortia. And this is not public yet, but if some of you are interested in using this collection, please let me know, email me or whatever, and I can pass you the contact of the person that is responsible for this collection. Multiple people are responsible, but you know what I mean. So from this very, very large collection of sediments, we needed to select a few in which we could perform our experiments. So in order to do that, we based our selection in a couple of criteria. So one of them was either if it had a PCR, a MER-A PCR positive result, So if we found the MER-A, it was okay. Or if they were isolated with a pre-treatment of mercury chloride. So if before isolation, they had to endure a high concentration of mercury that we added, that was also okay, good. They can either have the gene or tolerate mercury. And also we take into consideration taxonomy based on previous literature, and also sample distribution. So if the species was found in more than one area or only one area. So taking all these criteria into consideration, we pre-selected 37 isolates and 7 consortia. So the paper that we published was only for the isolates. I'm gonna present the consortia as well because it's we did the work parallel, but the consortia will be published with something else. So if you have any further questions about the consortia, you will not find the information available in the paper, but you can just ask me. So for all of this 40-something candidates, we perform different physiological tests to see which candidate was the better one for a multitude of conditions. Among them, we tried salinity, so the ability for the candidate to grow in either a marine salinity or a brackish salinity. From the ones that grew well in both salinities, we did an anaerobiosis assay to test the tolerance to low concentration of oxygen. And also we did minimum inhibitory concentration with both inorganic mercury and methyl mercury. And from all of these assays, we selected 3 isolates and 1 consortia to go to the next part of the selection process. So the next part was to do growth curves to characterize the growth of the isolates in either control conditions, which were with no mercury present, or in 2 different concentrations of mercury. So this is, we selected 4 micromolar and 12 micromolar, which is basically 1 ppm and almost 3 ppms. And also we did a negative control for the abiotic degradation. So we selected 1 ppm and 3 ppm for the growth curves, because even if the concentrations in the environment are higher, not all the mercury that is present in the sediment is available for the cells. But here, because it's liquid solution and just mercury chloride, everything will be bioavailable. So we wanted to take this into consideration, and that's why we chose lower concentrations of mercury than the ones present in the environment. So in order to follow their growth, we took OD, absorbance samples. So then we could measure the growth rates. And also we took measurements for total mercury to be measured with the AMA 254 to calculate the percentage of removal of mercury. So the first isolate that we tested was an Aeromonas species, and even in the control conditions, we saw that they did this kind of weird S in the growth. This is due to the fact that they make very large filamentous aggregates like this one. I don't know if you can see it very well. This is the best picture that I have, but it's still not very good. And when we added the 4 micromolar of mercury, we saw that this curve was even more pronounced. Curiously, when we added 12 micromolar, we did not see this curve. And also we see a difference between one of the replicates and the other two of the replicates. So two of the replicates have a lag phase of around 20 hours. And then the one replicate that is the square one of the replicates didn't have any lag phase at all. So my first thought was, okay, so we forgot to add mercury to that sample. But then we measured the mercury and that was not true. All of them had mercury, so we could not really explain why one of the replicates was a lot faster in degrading the mercury than the other two. When we see that in the 4 micromolar, we see that there is almost 80% of removal in the first 24 hours, but we see in the curve that this is 2 hours, 4 hours, 6 hours. We see that basically is the lowest point of the lowest concentration of mercury is already at 2 hours, 6 hours. So we see that there is a very good degradation rate in this isolate. And also in the 12 micromolar, we have to take into consideration the differences between triplicates. But even taking that into account, we saw almost 54% of removal after 24 hours. While in the abiotic control, we only saw 35% of 46% removal. So it's 20% more of what we saw in the abiotic. So the next one that we did was the Pseudomonas species. This one had was very consistent among replicates. We didn't see any weird things like in the previous one. The growth rates in the previous one, the growth rates couldn't be measured because they made aggregates. So the measurements weren't good enough to be able to measure the growth rate. But in this one, we see that the growth rate is basically the same under the 3 conditions. So it doesn't matter that it has mercury or not. The growth rate is basically the same. But we observe a lag phase in the 12 micromolar of around 22 hours or so. When we check for the degradation, we see that the percentage in the 4 micromolar is basically the same one. But in the 12 micromolar, we were expecting for it to be lower after 20 hours, after 24 hours, which is basically what the lag phase lasted. But it was not. It was a steady at like 6 micromolar, which was already higher than the previous condition. So our working theory is that some of the degradation at the beginning was done abiotically, and then when the cells started to grow, they started to degrade as well. And yes, we needed to take longer times to see the full degradation. The third one that we tested, so these two, the Aeromonas and the Pseudomonas, were selected because they had positive MER-A PCRs. And this one, which was a Bacillus species, was selected because it was obtained from the pre-treatment with inorganic mercury. So we knew that it was able to tolerate high concentrations of mercury. So we tested it, but then when we tried to measure the growth, we saw that the samples made no sense, like they didn't follow any pattern at all. And this is because they made these very large cloud-like aggregates. So we tried to break them down, but there was just no way. So as you can see, the cells are growing and they are growing just fine, but because of the aggregates, we couldn't measure it. So we thought, okay, maybe they are just like degrading it the same, but we couldn't measure the growth. But then when we measured the mercury, we saw that the concentrations were steady in the 4 micromolar, It was in the abiotic was 10% of removal of loss after 24 hours. And we see that in the bacillus condition at 4 micromolar, the percentage is only 11. So it's basically the same one. The cells must be using some other mechanism in order to be able to tolerate the concentrations of mercury instead of Med-A. So this one could not be used for bioremediation. Additionally, this is the part that is not on the paper. So if you have any doubts, you can ask me at the end or by email. So we tested also a consortium. This consortium was composed of 3 species, one Marinobacter, one Talosyspira, and Rosevium. And it was checked also in the 2 concentrations of mercury. Under the low concentration, we saw that the growth rate was virtually the same that in the control, but in the 12 micromolar, we see that the pattern has a very lag phase, very large lag phase. And then at the end of the experiment, this is almost 70 hours. It may be looking like it's starting to grow. And we see that the concentrations of mercury are also starting to get lower. So probably we would need more time to observe the full growth curve, more than 72 hours. That was what the experiment lasted. But we see a very good condition, but very good degradation curve for the 4 micromolar, reaching up to 74% as well. So the percentage were pretty much the same, but in the 4 micromolar. But we see that well, except for the bacillus that didn't degrade at all. But the Aeromonas did very large aggregates, which could mess up the next step. So we decided not to do it. So the next step, not to use it. The next step that we did in order to optimize the conditions for degradation was perform a continuous culture experiment. I'm gonna explain a little bit of the theoretical part of the continuous culture. So basically what we have is a bioreactor in which we enter fresh medium at the same time that we exit dirty medium with cells on it. So basically, by setting a velocity, a dilution rate, what we can then set, this is the equation that is used theoretically, but basically the concept is that if you leave it long enough, the whole container of the bioreactor will reach a steady state in which the number of cells will be constant and all the cells will be growing at the same rate. So basically, but setting the dilution rate, you can set the growth rate of the cells. So we decided on 3 growth rates that we wanted to test for the 2 candidates that were more promising. And that's 0.05, 0.1, and 0.2 hours -1. So this is an actual picture of what the setup looked like. So this is our bioreactor, this is our fresh medium, and here at the, like on the floor, there's a large container of just waste. So this is the growth rates, 0.05, 0.1, and 0.2. I'm gonna be talking about these numbers, but what you tell the pump is like, okay, so you need to enter 0.82 milliliters per minute into the bioreactor. So the cells can grow at 0.05 hours -1. So as you can see, this is an very large, this is 13 liters of medium, and it's also an open system. So this is because the cells need to be exiting continuously. This is an open system, so we were not able to use mercury inside the system. So there was no mercury neither in the fresh medium nor in the bioreactor due to safety, laboratory safety. And also we could not assure that the concentration that would be entering the media, the bioreactor, would be constant, because it could be absorbed by the bottle or by the tube. It could be photo reduced. So because we cannot change the conditions of the bioreactor, we decided that once we reach the steady state, we were gonna stop the experiment, divide the 1 liter that was in a steady state inside the reactor into 3 replicates, and then spike those with 4 micromolar of mercury. So in order to corroborate that the degradation of mercury was taking place the closest to the steady state that we could achieve, we took, as well as mercury samples, DAPI, so DAPI samples for counting the number of cells that we had. And also we wanted to check the expression of the meta-A, and also we took RNA samples and performed digital PCR. This was done for the 2 candidates that were more promising, so for the consortium and for the Pseudomonas. The consortium was different for the slowest time than for the 2 fastest, for the fastest and the intermediate time. So we see that there is an effect on the number of cells that we have due to the dilution rate. And we see that this effect of dilution is much more pronounced in the Pseudomonas. So in the slowest time, we have a 1.2 absorbance in the Pseudomonas, but in the highest growth rate, up 0.2, we only have 0.37, 0.4 of OD. So we have a much lower number of cells in the fastest time than in the lowest time. So because we have the number of cells and we have the concentration of mercury, we can basically calculate how much mercury there is per cell. So we see that in the consortium, all the three steady states were pretty much the same. But when we when we measure the Pseudomonas, we see that the fastest time, so the 0.2 growth rate, had much more mercury per cell. So the concentration of mercury was the same, but the number of cells which was much lower. And even with a higher concentration of mercury per cell, the concentration after 2 hours was basically the same one. So we see that they are degrading much faster at the highest growth rate than at the slowest growth rate. This concentration of both cells and mercury was used to calculate the specific degradation rate. So picomole of mercury per cell per hour was what was disappearing from the bottles. So in the consortium, we see that basically the numbers are the same, 5.5 or so picomole of mercury per cell per hour. It didn't have, the growth rate didn't have an effect on how much mercury the cells were degrading. But when we search for the Pseudomonas specific the degradation rate, we see that in the smallest time, it's already 10 picomole of mercury per cell per hour, which is already almost double of the consortium. And we see that at the highest here's this day, but it's not day, it's hour, sorry. At the highest growth rate, it's almost 6 times more than the consortium. So decision time came. We needed to say, okay, what's the best for detoxification of mercury. So with the consortium, some issues arose. One was that it could be less stable over time, because it was a combination of three cells of three different species. We were worried about one of them losing viability, viability, or even inside the reactor, one could be washed out. So we weren't completely confident on the stability of the consortium. Also, we were not able to improve their degradation capacity. And also the species that were found in the consortium had less literature on them. Instead, while the Pseudomonas was much more common, so there were many more studies done on different species of Pseudomonas. It was found in a multitude of environments. So this could potentially open the door to not only bioremediating sediments, but also maybe like waste waters or waste from different things, or even soils. Some people in Brazil use Pseudomonas to, not this one maybe, but we don't know. We could potentially use it for more environments. And also we could improve the degradation sixfold by growing it, sixfold compared to the consortium, by growing it at a high growth rate. So the best candidate was Pseudomonas. I don't know if you're seeing it blurry. I see it blurry for some reason, sorry. In order to check that that the degradation of mercury was done by ourselves, we check for the expression of the MET-A. And curiously, we found that the highest expression of MET-A was found in the intermediate state, so in the 0.1 instead of the 0.2. We hypothesize that this is because we had insufficient time to resolution. So we know that the RNA can change in a matter of minutes. And maybe what we think that happened is that we just missed the peak, or that we, for some reason, the RNA in the highest growth rate was degraded. But yeah, we cannot, so these results differ a little bit from what we observe in the concentrations of mercury. But what we can see is that even at different ranges, because the axis has different has a different scale, we see that in the steady state, which is the SS, there is no expression of MET-A, or very, very low expression of MET-A. And then at time 0, when we added the spike, the concentration, the expression of MET-A was triggered. So this was already very cool to see. Additionally, we did sequenced the genome of Pseudomonas and found one operon with only MET-A. It had not MET-B. And also we found other metal tolerance genes. There was a complete arsenic operon for arsenic reduction, and then also partials of gold, cobalt, zinc, silver, and some others. As well, we found some antibiotic resistant genes in which we need to be careful. We did a antibiotic test, multiple antibiotic resistances, to check that it would not interfere with the environment. But yeah, we need to be aware of how this could potentially affect the environment in which you will be using it. So I'm already almost done. So the best take-home messages are Pseudomonas is the best, and growing it at a high growth rate is the best operational conditions. So Pseudomonas was able to grow in a multitude of salinities, in multiple salinities. It was able to tolerate anaerobiosis conditions, had a high tolerance for mercury, and also a high percentage of removal. And we were able to optimize threefold the degradation of mercury at a high growth rate. So thank you for your attention. I will take questions, and also wanted to thank everyone that was involved in the research, people from the university, people from the ICM, and also the Mercurius lab, which is Andrea's lab. And I will be ending my PhD in hopefully a couple of months. So I'm searching for a postdoc. So if anyone has open positions or knows of someone that is hiring a postdoc, please refer me to the position. So yeah, that's it.
Thank you very much, Carla. Bravo. Yeah, very interesting. A lot of things. It feels like, yeah, it was a lot of work.
Yeah.
So as Carla mentioned, if there is anyone who wants to ask a question, please go for it. Don't be shy. It's nice. Otherwise, I have questions, but I can't see anyone. I will start probably. People will follow. Thank you for taking the time to present your work, and it's super nice. I did not have the time to read your paper, so I'm quite happy, and I feel honored that I can get the special presentation. I was just wondering, like when you did the growth, you didn't speak about like which kind of media did you use?
So we used a kind of a modified Sobel medium. So we created the seawater. So we had a recipe. I can pass you the recipe if you want to. So a recipe for seawater, because we wanted to use the same one that we used for the salinity assays. And we needed one in which we could modify the concentration of salt. So we created the seawater, and then just use yeast extract and bactopectona to make Sobel.
Okay, just like then now I'm just wondering, like, so what is the final purpose of isolating a candidate that is super good of like who can resist mercury concentration and can remove it? I guess, is it like any way that it could be applied in water plant treatment, or and in that case, why will you choose to have like a media that is with salinity, like with like a could be like a marine water, if you are in a system that where most of the time we have fresh water?
So we tested for the salinity because we were planning. So the project was to bioremediate sediments, marine sediments. So we needed bacteria that was able to grow on saline environments. And this was, it was not as part of this paper, but tested in as part of the project. We did a pilot plant study with a very large tank of 2,200 liters. And in there we had sediments as well, sediments and water. So it was a slurry. And we saw not as promising of the results as we wanted. So the next step that we could take would be to use it for wastewaters. So I think that that's going to be an issue because we also tested low salinities. Of course, we will need to test. We will need, if we were gonna be using it for water plants or for wastewater treatment, we would need to test the operational conditions. But Pseudomonas is usually a great candidate for bioremediation. Many of the studies that I have searched for bioremediation are done with Pseudomonas exactly for this reason. They are good.
Cool. It's good to know. I think Jan has a question, so please go for it.
Yes, thank you. Can you hear me?
Yep.
Yeah, okay. Thank you for a very nice, very interesting presentation. I have a general question about the reaction product in your degradation step. So when you talk about degradation of mercury, I assume you mean the reduction of mercury 2 to mercury 0, and then the mercury 0 as a volatile species is assumed to leave the system. Do you have any control over this? Are you sure that all of the mercury that you didn't find in solution anymore actually was transferred to the volatile phase, to the gaseous phase by volatilization? Or could it also be that there are other immobilization mechanisms, that some of the mercury you didn't measure in solution anymore was maybe stuck to some surfaces? Or do you have any control about the product of the reaction? Thank you.
So this is a two-part response. So in my study, we did a abiotic control. So that had that bottle had no cells. So it's this one. A ver. Sorry. So this one had no cells. So it was basically to check exactly for that. So if it would be maybe attached to the bottles, or if it was lost on the sampling recipients or whatever. So this is in our experiments, this is what the abiotic control is for. And we checked and we saw that the loss that we had for abiotic reasons was in the 4 micromolar up to 10%, and in the biotic and in the 12 micromolar up to 35% approximately. So we know that at least 10% of the 4 micromolar is being lost for some other reason that has nothing to do with our cells. But in the pilot plant, so in the general procedure that we would use to bioremediate the sediments, the large bioreactor we have would have an activate carbon filter in order to prevent the mercury to leave the system. So it's not truly degradation, it's just removal from the sediments. So then we would obviously have to figure out what to do. Well, they have it figured out, but you will need to treat the carbon filters to store them properly and to dispose them properly. But we just, So our goal was to take it out of the environment, so it couldn't harm the fish and also like the people that live there. So it was not really degradation. It's just, yeah, reduction and then treating the reduced mercury in some other way.
Okay, thank you. Yeah, so but in your bottles, you didn't have a mercury sink then. So then the mercury was volatilized and maybe stayed in the headspace as mercury vapor.
Yeah, what I think is happening is just in the headspace, and then we because we have to open the bottles in order to take the samples. So even if we wanted to check the headspace, it wouldn't be possible with this setup. We would need some other setup in order to just check the headspace. So yeah, probably it's on the headspace, but we have no way to check it for that.
Okay.
But we assume that that's what's happening.
Maybe last detail about this. When you measured the mercury concentration, you took an aliquot of the suspension.
Yeah.
And so this included also the biomass.
Yeah.
So basically what you measure there is a mixture of the solution and the biomass. So if there would be binding of mercury to the microbial cells, you would still measure it there.
Yes.
Which there probably is. Because in the abiotic control, you don't see mercury sorbing to the biomass. But if it's going down, then of course it's not just sorption, then it must be going to the vapor phase. Okay.
So yes, that's what one of the things that so when I started the experiment, I was expecting to have an end concentration of 0 mercury, like no mercury at all. That didn't happen in any of the solutions, in any of the states, in any of the experiments that I did. So I'm assuming that some of the mercury, it's binding to either the biomass or just it reaches a concentration in which the cells can just transport it out without using too much energy, and just it's not worth it to have the MER-A expression anymore, or the concentration of mercury is so low that the transcription of the MER-A is not responding anymore. So it could be a couple of things, but yes, I am assuming that some of the mercury is attached to the cells. But because I want to see the mercury removal of mercury as a whole, I didn't differentiate it. One of the things that I wanted to do with this species, with the one that made aggregates, like, so for example, if I would do an experiment in this strain, I could take the biomass and solution separately and measure what's happening, if the mercury is soluble or if they are using like specific proteins to trap them on the aggregates, because I saw that this is a possible mechanism that they may be using. So for example, in this one, I could treat them separately if I was doing the experiment again. But in general, I didn't care. I just wanted to not be there.
Okay, thank you very much. I'm sure other people have questions as well.
I have here Judith, who started her PhD at Stockholm University, and she will also work a bit with microbiology, microbes. Yeah? And apparently she has a question for you, so I will leave her.
Yes, so hi, and thank you for the presentation. My question is more quite a broad or generic question. So on the last slide, one of the last slides you were showing with the genome information of Pseudomonas, you mentioned that it had, for example, a lot of antibiotic resistant genes and multiple resistance genes. So I was just wondering how viable would you think that Pseudomonas would actually be a good candidate for bioremediation? Like, we know that it really has this high mercury removal capacity that you showed, but then if it also introduces antibiotic resistant genes into the environment. So I'm just wondering, do you think it would actually be a practical solution, or do you have some propositions to overcome these other ethical drawbacks, I would say?
So yeah, this was one of the things that we were worried about. We wouldn't be introducing any new resistances because this Pseudomonas was isolated from the sediments. So the resistant genes are already there. We would be biomagnifying these specific species. So that could be an issue if the cell were pathogenic. So we check for some physiological traits of pathogenic indication. So we group them on agar blood and also check for some of the genes that are responsible for the most known pathogens. And we didn't find it. So we had no signs of these species being a pathogen. So this wouldn't be an issue on terms of creating or biomagnifying a possible sick possible pathogen for the fish or algae or whatever living in the space. But because also we wouldn't, like, because in the pilot plan, we saw that the bioremediation of actual sediments was going to be much more complicated than we hoped. And also, I didn't explain this part of the paper because it's a very complex procedure and I didn't do it. Some people in Germany did. But we perform immobilization for the so in different in like small pieces of rocks. So what we wanted was to add the rocks, and then before putting back the sediments, removing all the rocks with the cells on it. So this is one of the things that we try to do for the pilot plant. So in theory, the cells wouldn't even be on the sediments after bioremediation, but obviously some of the cells would still be present. But yeah, this is one of the issues that we had to deal with. So our response was to search for the pathogen indicators, which we didn't find. So in theory, it's not even an issue, but still it could be developed or transform or mutate or whatever. So it's still something that we have to take into consideration when applying these solutions. It's just one of the factors. There's not really a way around it.
Okay, thanks. Thank you. Anyone has a question? It's the time. You won't have this opportunity unless you go to South Africa for the Mercury conference. I don't know.
I will not be going to South Africa this year, sadly.
Aha,
So
this is the time.
this is the time. It's now or never. No, that's not true. You can email me whenever.
But I mean, if there is no more question, I don't see the point of going further. But first of all, I would like to thank you again, Carla, for your very nice presentation. And I would like to thank you all who came here and make this seminar happening too. I would like also to let you know that in April, we will have like a discussion seminar coming, and it will be about speaking about the gene AGCA. So I'm just like bringing the gossip here for the people who are here. You will have some information in the coming weeks, but yeah, we are trying to work a bit to make some nice thing and nice discussion coming. And of course, if any of you are interested to make a presentation or has idea or want to share also the link to be part of Mersorcium community, please feel free to make it. And it would be nice to have a new members. And that's it. I just wanted to thank you all and thank you again, Carla.
Yeah, no problem.
Yes, very nice. I'm super happy that we did
As
it.
always.
Very nice. And good luck for the next step. For finishing the